Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Essay The Krebs Cycle, a Critical Examination. The year 1937 witnessed a landmark contribution to the landscape of biochemistry, Hans Adolf Krebs' delineation of the citric acid cycle, subsequently known as the Krebs cycle or the tricarboxylic acid cycle. This essay undertakes a meticulous examination of Krebs' seminal work, focusing on the intellectual contours of his claims, the methodologies underpinning them, and the enduring impact of his discoveries. The overarching aim is a critical assessment of the cycle as it was presented within that pivotal year, scrutinizing the logic, empirical support, and conceptual innovations that defined this groundbreaking contribution to the understanding of cellular respiration. Section 1 Introduction. The genesis of an explanation. Krebs' work, primarily stemming from experiments conducted on pigeon breast muscle, sought to elucidate the mechanisms underlying cellular respiration. Prior research had made headway, but a comprehensive understanding of how carbon-based molecules were oxidized to generate energy remained elusive. The existing knowledge base comprised studies such as the investigation of oxidative phosphorylation, the understanding of glycolysis and the role of different enzymes. The central task was to comprehend the intermediary steps through which carbohydrates, fats and proteins are broken down and transformed, ultimately yielding usable energy. Krebs' exploration offered a systemic framework that explained these mechanisms through a connected cyclical process. This essay will concentrate on the claims Krebs proposed, his supporting evidence of the implications of the work from a critical perspective. Section 2 The discovery of the cycle, a systematic approach. Krebs' central claim was the existence of a cyclic pathway of reactions, a pathway that would become known as the citric acid cycle. His method involved the systematic study of metabolites in respiring muscle tissue. He observed the impact of various organic acids on oxygen consumption. He showed that the presence of specific dicarboxylic or tricarboxylic acids, such as citric acid, significantly increased the rate of oxygen uptake. This observation became the cornerstone of his argument. Krebs' method involved carefully manipulating the concentration of various compounds and monitoring the resulting oxygen consumption of the pigeon breast muscle, measuring the formation or disappearance of specific compounds. He deduced that the cycle began with the condensation of a 2-carbon molecule acetyl-CoA with a 4-carbon molecule oxaloacetate to form a 6-carbon molecule citrate, which then underwent a series of transformations. The cycle proceeds through a series of enzymatic reactions, each carefully catalyzed and each resulting in the regeneration of the starting molecule, oxaloacetate. This regeneration explains why the presence of even small amounts of the cycle intermediates, such as citrate, could substantially stimulate respiration. The evidence provided showcased a tight coupling between oxygen uptake and the addition of the specific acids. These observations formed the critical basis for his description of the cyclic pathway. Section 3 The identification of intermediates, a chemical perspective. A core component of Krebs' model involved identifying the specific intermediates involved in the cycle's various transformations. He carefully measured the changes in concentrations of various compounds over time, both in the presence and absence of added substrates. Through meticulous experimentation, Krebs was able to show the production and consumption of several key intermediates, each a crucial player in the series of reactions. These intermediates included citrate, cisaconate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. The appearance and disappearance of these substances provided direct empirical evidence for the sequence of reactions. He demonstrated, for instance, the formation of alpha-ketoglutarate from isocitrate, identifying a key oxidation step where carbon dioxide was released. Similarly, the conversion of fumarate to malate via hydration was a crucial step along the cycle. His focus on the role of succinate dehydrogenase, an enzyme that facilitates the conversion of succinate to fumarate, provided an additional data point. The evidence was simply the identification of intermediate and demonstration of their sequential transformation, thereby defining a pathway connecting various carbon-containing compounds. Section 4 Experimental Validation Physiological Significance. To buttress his claims, Krebs integrated his biochemical findings with an understanding of the physiological context. He demonstrated that the key steps within the cycle were sensitive to the presence of specific inhibitors. For example, the inhibition of succinate dehydrogenase, a crucial enzyme, led to the accumulation of its substrates. He integrated that biochemical reactions were directly linked to cellular respiration. This provided additional evidence supporting his cyclical model. He also carefully considered the role of the cycle in generating energy-rich compounds, particularly the production of reduced nicotinamide, adenine dinucleotide NAD, and reduced flavin adenine dinucleotide FAD2. These molecules, later understood to be essential for oxidative phosphorylation, were shown to be generated during several steps of the cycle. The generation of these molecules proved the pathway had far-reaching consequences, directly linking the cycle to the complex system of energy production in biological organisms. Such findings helped to unify the biochemical aspects of the cycle with its physiological relevance. Section 5 The importance of the coenzymes, the catalytic role. Krebs, in his work, devoted attention to the involvement of coenzymes. He did not possess the full understanding of the specific coenzymes and their roles that is understood today, yet he recognized their essential contribution to the process. While the identity and precise mechanisms of action of these coenzymes were not fully understood at the time, their importance was clear. Krebs proposed that the cycle relies on the presence of several coenzymes to facilitate the enzymatic reactions. The experimental observations that oxygen consumption was dependent upon the presence of oxidized intermediates suggested the participation of electron carriers. This indirect evidence provided by Krebs established an early framework around the coenzymes, setting the stage for its eventual characterization. It also opened the eye to recognizing the role of ATP in this process, which would be discovered shortly after. Section 6 The role of oxaloacetate, a catalyst's crucial function. Krebs insightfully highlighted the pivotal role of oxaloacetate. He recognized that this molecule acts as a catalyst, continuously regenerated at the end of each cycle. The realization that oxaloacetate acts as a catalyst was a fundamental insight. It provided an explanation for the observation that even small amounts of intermediate could stimulate entire process. The cyclic regeneration of oxaloacetate was crucial in Krebs' understanding of the cycle. The molecule is regenerated at the end of the cycle, ensuring the continuous flow of reactions as long as energy is available. This insight was a crucial departure from the understanding of metabolic pathways that were the prevalent scientific undershare. Section 7 Comparisons and Connections Positioning. The cycle to existing knowledge. Krebs' work, while innovative, emerged within a complex intellectual landscape. Earlier experiments had laid the necessary groundwork for understanding cellular respiration. For instance, Otto Warburg's studies on respiratory enzymes and carbohydrate metabolism had established many of the foundational concepts. Krebs' cycle, though, was a distinct advancement. One key difference was in its emphasis on organic acid metabolism. He explicitly connected his experimental findings with the existing knowledge of glycolysis, notably the conversion of pyruvate, generated via glycolysis, into acetyl-CoA. The molecule that enters the cycle was a critical juncture linking these two fundamental metabolic pathways. His articulation offered a cohesive framework that connected diverse pieces of existing knowledge. This approach was key in establishing the validity of the cycle and securing its acceptance. Section 8 Methodological Strengths and Limitations and Empirical Assessment. The strengths of Krebs' approach lie in his experimental design. He focused on measuring objective parameters. He studied well-defined parameters, such as oxygen consumption, by employing detailed control. The careful choice of experimental conditions, such as concentrations of added substrates and the use of muscle tissue, provided clear and consistent results. However, The methodology also had limitations. The technology of the time limited the ability to precisely study the enzymes involved. The techniques to identify and measure intermediate compounds were less sensitive. He lacked the tools to fully resolve each reaction or to characterize the specific enzymes involved. While the cycle was proposed with considerable accuracy, refinements that come in later, Section 9 Reception and Refinement. The cycle's immediate impact. The reception of Krebs' work was swift and largely positive. The scientific community recognized the importance of these findings. His recognition with the Nobel Prize in Physiology or Medicine in 1953, along with Fritz Lippmann, for their discoveries concerning the citric acid cycle and the role of coenzyme A, provides proof of this. Subsequent research, building upon Krebs' initial proposal, added new layers of detail. This includes the identification of specific enzymes, the characterization of their mechanisms, and the elucidation of the precise roles of electron carriers. Many revisions and elaborations were made. The work refined the understanding of the cycle, but the fundamental structure that Krebs had proposed has endured. Section 10. Enduring influence. The impact of a revolutionary view. Krebs' discovery of the Krebs cycle established a foundational understanding of cellular respiration. The impact of his work is profound and is visible in the fields of biochemistry, cell biology, molecular biology, and medicine. The cycle is central to energy production in nearly all eukaryotic cells. The cycle provides a basis for understanding a wide array of biological processes, including how organisms derive energy from food and how metabolic dysfunctions contribute to human disease. The cycle's presence in numerous organisms, from yeast to humans, underscores its universal significance. The widespread impact of the cycle reveals the power of Krebs' method as a way to investigate and understand the most fundamental processes of life. Krebs' work stands as an important example of the power of scientific inquiry. Section 11. A critical conclusion reflecting on the value of the cycle. Krebs' 1937 work on the Krebs cycle established a framework for understanding cellular respiration. Krebs developed a systematic approach to study the intricacies of metabolism. His emphasis on oxygen consumption as a marker for metabolic activity, coupled with detailed observations of the intermediates formed, provided a firm foundation. Despite the limitations imposed by the technology available, his conclusions were remarkably accurate. His experimental design and dedication led Krebs to the correct identification of a psychic biochemical pathway. The cycle's enduring recognition in biology highlights the originality and significance of his contribution. His analysis showcases how careful experimentation and insightful interpretation can illuminate fundamental processes. The Krebs cycle, as presented in 1937, remains a benchmark. It reveals the enduring impact of scientific discovery and the importance of critically evaluating the framework and claims put forward by scientists.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.